you who have come and spared time to come and listen to me. I must thank you. This is an overwhelming feeling in my... I have gone to many universities, given lecture, but this is an outstanding place. So I thought I'll talk about vaccines. And this is an area in which I have researched in, but when I started looking at it, different vaccines, pathogens, how they are made, what is the science behind it, it's a very large subject. So I'll say right in the beginning that I'll try to address to all of you, not as a scientist, but something more general. So I'll make some few statements which are general statements, and that scientists will excuse me for making generalized statements, but the social science will excuse me if I use something more technical than you will understand. Very large number of people died in the First World War in ditches because they had bacterial infections, and there were no vaccines at that time, and they had viral infections. Spanish flu killed more than 50 million people, documented 50 million people. The number will be even more large. Today, that kind of numbers are unimaginable because neither we knew the viruses at that time, nor we knew how to protect ourselves, and that happened only about 100 years back. So successes of vaccines are reflected, familiar. In fact, young generation would not even know what is smallpox, because when I was child, we were vaccinated with smallpox. So one of the major success of vaccine, one can call success, but the virus is not great success. In the sense we almost removed as much as we know, we removed smallpox from infecting anyone. So there's a great success. Polio, for example. When I was a kid, you would see a lot of children with polio. Today you won't see young or older people becoming disabled because of polio. Polio vaccines managed to do that. And I'll take COVID as an example of vaccines, because all vaccines, all viruses are complex. All viruses look similar in their construction. Yeah, three general statements I forgot to make. Let that slide be there. I was saying that this is the body, where this is a complex human machine. It protects us from very large number of infection diseases. So we have innate immunity. That's the first layer of immunity. The skin is there, protects us from many diseases, infections. That's given. But there is something called acquired immunity. When you are exposed to a foreign material, if that is infective, then you acquire immunity over the time. So that's acquired immunity has two arms, and that will, in passing reference, will come, so I thought I'll just get it out of the way. One is you raise what you commonly hear, antibody response. So that's if these are two arms of immune system. One is majorly, which is any foreign material going in, body responds very quickly to raise an antibody response. Many diseases were diagnosed by the presence of antibodies, presence of pathogens, but also presence of antibodies. The other arm is what is known as cellular immunity. Just live with this word, cellular immunity. These are generated by a cell system called B cells. We call them B cells, and these are from T cells. So this is one arm, this is the other arm. These both arms communicate with each other. Without robust T cell responses, you cannot have B cells producing antibodies. So when we are exposed to a pathogen, these two arms get into action. And there are a large number of cells, I won't name them, but there are many cells which get into action. And this is because we evolved over so many years. So we have a great defense system. But this great defense system is very susceptible because out there are lives which depend on infecting. It's only in 1917, almost beginning of the 19th century, when the first vaccine, great-great-grandfather of vaccines, Edward Jenner, in your school books you would have read about smallpox vaccine. That's just observation. These scientists were not even called scientists at that time. They were almost, I would call, social scientists who were great observation power. He was a doctor, but a great observation power. So the first vaccine, which was a live vaccine, this word will come, live vaccine, attenuated vaccine, killed virus vaccine. It simply would mean, live would mean that there's a vaccine which will be given to us as a live pathogen, small amounts of live pathogen, and then body would respond. The danger of live vaccine is if you have not responded very well, then you might even get the disease. But this is how the initially live vaccine, the vaccine began. It took almost, you can see, 100 years, almost 100 years, between the two, when the anthrax is a deadly disease, killed millions of people. Anthrax vaccine is the first vaccine that was created in a laboratory. It's a bacterial infection, and you isolated the bacteria, grew the bacteria, and killed it or attenuated it. So attenuated vaccine would be when you take the pathogen, but make it weak to such an extent that it can't cause infection to you. But for all practical purposes, in your body, the pathogen is going. So the body would respond to pathogen regardless, whether it's live or not live. And then very big change, rabies vaccine. Rabies was such a disease with the cats and dogs, and no protection. And I don't, the lecture gets very long if you start talking about diseases, but rabies, as you know, needed to be controlled. So rabies vaccine came in 85, and then another 50 years, the BCG, the tuberculosis is one of the biggest killer in Europe, everywhere, Europe, wherever. A lot of people died at very young age. They were isolated. There were no cure for TB, no drugs, of course, for TB at that time. So the BCG was great landmark in vaccine, history of vaccines. So between 1920, or let's say 20s, this is just after the First World War, and then go on to the next World War, when the structure of DNA was revealed to the world, that people knew their DNAs are there much longer, much before, but we didn't know how DNA multiplies. How does it like an auto machine replicates itself? But when the structure in 1953 at Cambridge was given to us, that was the first time, the wow movement of biology, that you understood the basis of life, which is DNA. How would it multiply? We had no idea till only about not even 100 years back. So the science that we are discussing, or we will discuss, is really in a very short time. And after 1953 to 60, then biology simply exploded, because you understood how the DNA and RNA, that you read in the books, the dogma of RNA, DNA, and proteins. So this is another general statement, that our body is maintained, or all, for that matter, living bodies are maintained by the workhorse of our biological molecules called proteins. So when I raise my hand like this, I get some energy. Many enzymes are involved in pathways to give me energy to even say this or to talk. So proteins are working all the time. Smaller life forms will have lesser number of DNA that transcribe into proteins. More complicated, complex form will have what we call bigger genome, therefore larger number of bigger DNA, larger number of proteins. And by the time we come to humans, I think there are more than 20,000 proteins which are supporting this body. But when you start going back to the pathogens, which I believe, as a scientist, I believe must come into existence much before this form of animal, would be just keep going back, the genomes become smaller and smaller and smaller. But I'm not talking plant sciences, some of the plant science, genomes are very big. So I'm just looking at the life forms right now. So if we go to the most rudimentary form of life, or the information for that will be viruses. In fact, scientists are debating even today whether the virus is a living species or it is non-living species. We won't get into that controversy, but there are viruses, and I'll show you some pictures against which vaccines will be made. I took a break here because from 1920, the stories I tell you, till about 80s, very large amount of research was done because biology was entering. Many vaccines, which we just say vaccines are there, were actually discovered, and most of that work was done in places like, mostly in America, but also in Switzerland, France, and so on. But that provided us very large number of vaccines. Today, as I speak with you, it's in my slide, but I'll get it out of the way now. There are more than 30 vaccines which are available for us for immunization against various diseases. And when the policy comes, I'll let you know which are the vaccines which are fundamentally important as a child should have. So, by the time, as I said, molecular biology is affiliated, by 80s, we knew to break a DNA at will. We had enzymes and seal them back. So we could take a bacteria. I'm jumping now from viruses, but bacteria. Bacteria multiply, but they don't need any other cellular. They have complete body themselves who can reproduce. Just give bacteria a bread piece or sugar or right condition, it will grow, explode. By this time, scientists had figured out how to cut open a bacteria. And this was most of you know, this is how insulin is made now for the past 30, 40, 50 years. We just take a gene of insulin, insert in a bacteria, let bacteria multiply. It produces itself, but also produces insulin. We isolate, and insulin is easily available. Before that, insulin, if you need it, came from ox. You had to kill an ox, and that was not a human vaccine, that's closest to, and so on and so forth. But 1986 was the first birth of a lab-made recombinant vaccine. And this came just in 30 years of ability for scientists to understand and have a handle of molecular, what is known as molecular biology. Biology at a molecular level. If you understand that, and as I said, that DNA has all the information, proteins do all the work. There's one more class of biomolecules, and relevant because a couple of vaccines, most vaccines are against proteins of the pathogens. But there are exceptions, minor exceptions, I should mention that. The sugar molecules, which are glucose, sucrose, multiply, complex sugar molecules, also are extremely important biological molecules. So if I put DNA, RNA, protein, sugar, and lipids, I think then there are small, even sodium-potassium imbalance can kill us. So even ions are important. But as a large molecule system, these four are important. There are two vaccines, which we already have, are not against proteins. They are against sugar part of it, and maybe one of them, if I have time, I'll mention. So vaccines will be produced, can be produced. Future vaccines will always look at some part of the pathogen. So that was a fundamental when hepatitis B vaccine came up. And there's some politics here, economics here. Earlier, hepatitis B vaccine in India simply either wasn't available in Africa, India, or was very expensive. But vaccine was there. Production can be done. And I think India has a special place, and I'll mention that specially. In 2006 was extremely important, highly successful, very safe vaccine against human papillomavirus. You may not have heard of papillomavirus. It's been around for decades, for centuries. All viruses have been around for centuries. But in India, this was the most relevant vaccine, or is the most relevant vaccine, because in India, the load of women dying of papillomaviral infection, but infection didn't give fever, but it caused cancer. So in some sense, this was the first prophylactic or protective vaccine against cancer. So papillomavirus causes infection, but is the cause of cancer, mostly in women, but it can infect men also, but largely women. So women life, which were painful deaths, which was happening at various ages of life of a woman, can be completely stopped now. And HPV vaccine, again, in an Indian context, I'll have a reference. And finally, 2023, Prakash and I work with malaria. There is a malaria vaccine which I wouldn't say is available in India, is available worldwide, but has a WHO approval. This vaccine has come into being in 50 years. The vaccine work on malaria, because of problem of malaria as being such a deadly disease for young children, has been around from the 1980s. My own lab got involved in this in 1992, and we worked almost 25 years to get one vaccine in a phase 1 trial. But this vaccine developed at Oxford. All malaria vaccines are really based on a couple of proteins so far. And malaria has more than 5,000 proteins. So when those who are not scientists, what does scientists do to make a vaccine? You first understand the pathogen. And now that's not so difficult. For 50 years back, it will be very difficult, A, to isolate, B, to understand. Today, you can, and I'll come to COVID story. Let me come to COVID story quickly, otherwise this will become very long lecture. As I said, over 30 vaccines are available in the market for us to fight pathogens. Okay, I promise I'll talk about viruses, so I just say the viruses, all viruses contain either RNA, DNA. So the DNA virus, you have a couple of examples here. DNA virus like smallpox or herpes virus is a DNA virus. That is the virus, the piece of virus is made up of DNA. All viruses, when I say DNA, RNA, have a coat. They are protected. Outside there's a coat, all of them. Some are glycolated sugar, but the coats are made of proteins. So you might ask where the proteins come from. I am the virus, I multiply, where do I get these proteins from? If I take human proteins, of course I will not survive because body will get rid of me. So in the piece of DNA or RNA, in a viral, nonsense DNA you can make in the lab, it won't multiply. There is a special information should be there for multiplication. It can multiply, but it will have no meaning. So the viruses will contain, small virus, large virus, doesn't matter, will contain information to protect itself. After the virus genome has been made, it will cover itself. So there's always a coating on viruses. So that's the general mechanism. But they cannot replicate. They would lie around, hang around in the air or water or wherever, but they have no machinery. They need a host. So I'll come say the host could be plant, host could be tobacco, for example, can be tobacco mosaic virus will, tobacco plant, viruses, tomato viruses. Viruses would be, in fact, dogs, pigs. The pigs are a great reservoir of viruses. We don't quite understand why pigs are such good. Horses are infected by virus. So virus will find it. And there are specificity. What is fascinating is there are huge specificities. Some viruses will only go to the birds. And from birds they won't immediately jump. And I think I'll come to the COVID story when this jump start taking place from one animal to another animal and to the human, you start having problems. HIV is a famous example, which probably came from green monkeys in Africa. I mean, virus would be living happily in monkey for decades, for centuries. But the human contact, when it comes, it finds right opportunity. This is called the jumping of species. And then we are not ready for HIV, and then HIV gives us, of course it multiplies, Virus job is only to multiply. Job is not to infect or make us dead or give us disease. In the process, if that happens, so be it, and that's what happens. So it does have no machinery. It can anywhere. It can infect any living system. As I said, viruses are different shape, different sizes, different proteins they would express. I'll go to the next slide and just show you this is just some example. If you start from the left, small virus, poliovirus, and you say, how do you know this is small? How do you know this is this size? So this all is done. The genomes have been isolated. We know exactly how many nucleotides there are, what proteins will they make. You go a little bit up, you have adenovirus. Adenovirus is a kind of class of viruses which is also friendly. We can be infected with adenoviruses and not get, in fact there are now vaccines based on adenovirus, but adenovirus is the virus which also gives you prostate cancer, adenocarcinoma, but they're very slow growing, usually not considered very unfriendly to humans. Then you have a different shape, rod shape, tobacco mosaic virus. So if I can put many viruses, they're all look well different from each other. So I just thought pictorially, there's an Ebola virus and it looks like a snake. Ebola came into big light some years back in West Africa. It came from bats. So bat droppings would have the bats live heavily with the Ebola virus. Nothing happened to bats. They carry that business. Bats carry many viruses and nothing happens. So you might ask as a scientist, then what's so special in bats? Why don't we do what bats do? And that's a separate branch of science. So bats carry many viruses. So these are, as you can say, they're different size, different shape. All of them have information to multiply, and all of them have it covered. This is a little jerk. This is not a virus. I put it because both Prakash and I work with this person. Also give you some whiff. When we say malaria infection, malaria, what does it look like? How does malaria infect? Oh, that's a separate lecture. Next time when I come, we can talk about malaria. But when the infected female mosquito bites, the parasite goes in. Parasite gets into liver, we don't know what is happening in the liver, then it comes out. What you see on the slide here, and I wanted to show you a whiff of biology of the life which we casually talk about, but how fascinating, colors are given by artists or by us, but parasite in the bloodstream looks very much like what you see on the left. And you see a lot of information inside. This is absolutely correct information. This can be now actually seen by electron microscope. So science can open things that have never been opened for us. You can actually visualize, you can pictorial. I'll come show that in COVID virus also. So left-hand side is the parasite. You see the spikes coming out. These are not decoration. These are actually malaria protein. This is a pictorial representation. It on the surface of the blood stage parasite, there'll be very large number of protein. My lab worked on one of the major protein which makes the coat of this. Idea would be, if you see on the right-hand side, you see a red blood cell. Red blood cell doesn't get infected by viruses because there's nothing inside a red blood cell which will make you multiply. So virus actually in the red, we have a large number of red blood cells, doesn't enter. Virus doesn't enter red blood cell because inside the red blood cell there is no machinery. All you see is the hemoglobin. That's why it's a red color. It carries oxygen. So that's a fascinating part of being scientists and understanding biology. The virus also has no business, doesn't want to know because it's not going to multiply. But parasite does enter. So malaria parasite grows large amount in this dark blob of hemoglobin. And why I'm showing this, suppose you and I work on vaccine, what is our basis of vaccine? We'll say, all right, this is the first interaction, parasite is now trying to make contact. In the B, you see there's a fusion. This actually happens in real time. The process that I'm explaining takes few seconds. Maybe in one or 2 seconds, the parasite does this, red blood is there, right inside. But the scientists would say that let's not let the junction be made. What is the junction made up of? Which proteins are involved? Or, so if you can stop the parasite or by immunological method, that is antibodies or T-cell responses, then we'll stop infection. So that's the rationale. That's the rationale of any vaccine. Understand the pathogen. Today, you will not use the whole pathogen. You'll find, so the malaria vaccine that I mentioned in 2023, which is being now being given in Africa, is actually based on a similar protein from another stage of virus. So scientists now know a hell of a lot more, but I think we know so little that there's so much to be known. So I thought I'll just give you some whiff of biology of malaria parasite. This is how, and this is where the vaccines can be made up of. So there are live vaccines like smallpox, attenuated vaccine which will not give infection. Earlier polio vaccine were live and then attenuated. What we take today is attenuated. The other is killed vaccine. You grow the pathogen, bacteria, isolate it, kill it. You can chemically kill it, but you still will have all the parts of the virus or the pathogen, and you immunize. So these are the fundamental basis of vaccine.